Good morning, my name is Susana Nunes and our work is about predicting gene disease associations with knowledge graph embeddings over multiple ontologies. Experimental methods for gene disease associations are expensive and time-consuming to run. As a result, a number of computational methods have been developed to identify or predict gene disease associations. Methods that incorporate diverse datasets tend to achieve better results, especially for complex diseases, but obtaining associations on a large scale without compromising the quality of the associations remains a challenge. In this context, recent developments that explore the scientific knowledge described in ontologies have achieved good results. Ontology-based approaches for predicting gene disease associations include the more classical semantic similarity methods and, more recently, knowledge graph embeddings. The adoption of ontology for annotation provides a means to compare entities on aspects that would otherwise not be comparable. For instance, if a gene and a disease are annotated within the same schema, we can compare them by comparing the terms with which they are annotated. While this comparison is often done implicitly, it is possible to do an explicit comparison with semantic similarity measures. When biomedical entities are annotated with ontology concepts, we consider them to constitute a knowledge graph. Knowledge graph embeddings provide a vector-based representation of entities in the knowledge graph that can be used to infer relations. In comparison with semantic similarity, knowledge graph embeddings support in principle more powerful representations that consider multiple types of relations. Asif et al. Showed that machine learning classifiers trained on gene functional similarities using gene ontology can improve the identification of genes involved in complex diseases as was applied to autism spectrum disorder. Zhang et al. Used phenotype-phenotype similarity to prioritize novel gene-phenotype associations. These works focused on ontology and classical semantic similarity measures. However, these measures only consider hierarchical relations when measuring the similarity between entities described with ontology concepts. Recent advances in knowledge graph embeddings, such as Onto2Vec, EOPA2Vec, generate vector representations of biological entities and focus only on one ontology. To the best of our knowledge, Al-Sharhani et al. Built a knowledge graph with multiple ontologies but do not process gene disease associations to filter out possible cases of data leakage from DigNet. I will come back to this later in my presentation. In this work, we investigate the impact of employing richer semantic representations in gene disease association prediction. The semantic representations we propose are enriched because they are based on more than one ontology, they are able to represent both genes and diseases, and they consider multiple kinds of relations within the ontologies. We explored the human phenotype ontology and the gene ontology to represent gene and disease features in a shared semantic space that covers both gene functions and phenotypes. Human phenotype ontology provides a standardized vocabulary of phenotypic abnormalities encountered in human disease, and gene ontology describes the function of genes and gene products. We also investigate multiple knowledge graph embedding methods to represent both genes and diseases and how these vectors can be combined. We hypothesize that using more than one ontology can improve gene disease association prediction and that a richer integration can have a positive impact. In particular, we consider logical definitions that associate entities from different biomedical ontologies with complex semantic relations can be explored to bridge domains and contextualize relations between different entities, such as a gene and a disease. Here we can see the human phenotype ontology that has several logical definitions, including the gene ontology, and this allows to link both ontologies. For example, the human phenotype term for hearing impairment is equivalent to a restriction that involves the gene ontology term sensory perception of sound. Regarding our methodology, the first step in our approach is to integrate the different ontologies and annotation data to build the knowledge graph. Then we produce embeddings that represent the gene and the disease according to their annotations in different knowledge graphs. These embeddings are combined using different vector operators, producing a representation of genes and diseases in what is effectively a shared semantic space. Finally, supervised learning algorithms are trained over the combined embeddings to predict gene-disease associations. This approach is evaluated against non-machine learning approaches based on a baseline with semantic similarity measures and knowledge graph embedding similarity. Considering our dataset, first we obtain all curated gene-disease associations from DigNet. To avoid bias regarding annotations, we removed potential data leakage, considering only relations where the source did not rely on the databases UniProt, OMIM, or Orphanet. The resulting pairs were then filtered using ontology according to three criteria. The genes must have a correspondence with a protein from UniProt that is annotated with gene ontology terms, and the genes and diseases must be annotated with human phenotype terms. A total of 8,189 disease genes relations remained. Considering that negative samples are not included in DigNet, we employed random sampling to create negative examples composed of the genes and diseases present in the positive examples, but without known associations between them, building a balanced dataset. The knowledge graphs used were built with increased semantic richness and are divided in three possible types. First one, it's human phenotype ontology without the logical definitions and the human phenotype annotations for both genes and diseases. In the second knowledge graph, we added the gene ontology and gene ontology annotations for genes. The two ontologies are integrated to a common virtual root. In the last one, we added the logical definitions. Regarding the logical definitions, we simplified the existing ones to a more direct relations between the terms through an equivalent class. This allows for the path to be shorter and direct for random walk-based methods and for the other methods to extract a single tuple with the necessary information. The baseline aims to establish the performance of methods that use a single ontology and classical semantic similarity measures. We use the first knowledge graph, HP, since it annotates both genes and diseases, and measured semantic similarity for all gene-disease pairs using 6 different measures, 2 information content approaches combined with 3 set approaches. The association prediction is expressed as a classification problem where a semantic similarity score for a gene-disease pair exceeding a certain threshold indicates a positive association. For each measure, a semantic similarity threshold was chosen after evaluating the weighted average of F measures for positive and negative prediction at different thresholds and selecting the best one. The baselines represent the best possible result obtained with this approach. We generated knowledge graph embeddings with 200 features and used 5 different methods that covered 3 different approaches: translational distance, semantic matching, and random walk-based. Each gene-disease pair corresponds to 2 vectors associated with the gene and the disease. We define an operator over the corresponding vectors in order to generate a single representation. Several choices for the operator were considered, and the resulting vectors were then the input to 4 different machine learning algorithms. Grid search was employed to obtain optimal parameters to 3 of them. We also measured the cousin similarity between the vectors, carrying out the same approach used in the baseline. The classification performance was evaluated using the weighted average of F measures and AUC scores. In each experiment, we performed a stratified 70% training and 30% testing split method, being that the same split was used throughout all experiments, including in the baseline. Concerning the performance of the 6 semantic similarity measures using the first knowledge graph, BMA-Resnick achieves the best performance for a single ontology with 0.68 WAF score. There are three important aspects in elucidating the performance impacts of the different factors in richer semantic representations. First, how can we combine the gene and the disease vectors? Second, which knowledge graph embedding methods are more suitable for this task? And third, what is the impact of considering more than one ontology and logical definitions between them? Regarding the combination of gene and disease vectors, the three graphs show the 5-vector operations with the three best performing knowledge graph embedding methods coupled with random forest, one of the best performing machine learning algorithms. We can see the overall picture shows the Hadamard operator outperforms other operators, with difference being more evident when using op2vec. The following table presents the weighted average of F-measure scores obtained for the three knowledge graphs considered with the best performing embeddings and machine learning algorithms using the Hadamard operator as well as the cousin similarity-based approach. When using cousin similarity, the difference between the knowledge graph embedding based approach and the best baseline measure are reduced. However, when using machine learning algorithms, achieves better results than the baseline. On one hand, reducing the representation to a similarity score may be too limiting. On the other, knowledge graph embedding methods afford richer representations by both employing the full breadth of relations in the knowledge graph and providing multidimensional representations, something that semantic similarity measures do not. Furthermore, op2vec achieves the best results and is also able to take advantage of the integration of ontology with a small improvement in performance. op2vec generates a corpus with a set of asserted and inferred axioms from an ontology, the set of annotation axioms involving labels, description, synonyms, and creators, and the gene and disease phenotype annotations. By including all these features, this is a probable cause to present itself as a more stable and successful algorithm over the others. In conclusion, associating genes with diseases is an important area of research because it's a crucial challenge in human health with applications to understand disease etiology and develop new techniques for prevention, diagnosis, and therapy. In this study, we have proposed a novel approach to predict gene-disease associations using rich semantic representations based on knowledge graph embeddings over multiple ontologies. We investigated the impact of different approaches to build a shared rich semantic representation for genes and diseases, as well as multiple knowledge graph embedding methods and machine learning algorithms. We have shown that employing the best knowledge graph embedding methods and machine learning algorithm outperforms optimal semantic similarity measures by 8% weighted average of F-measure scores. The best knowledge graph embedding method was op2vec with more 1.5% compared to RDF2vec. However, RDF2vec, although less sophisticated, benefits more from using logical definitions than op2vec. Our experiments also reveal that the difference between using a single ontology or combining two ontologies, in this case human phenotype ontology and gene ontology, are comparatively small regardless of a richer integration using logical definitions. We hypothesize that the information provided by the logical definitions does not provide substantial additional information comparing with what is already present in the human phenotype ontology. This may be partially explained by the existence of only 351 logical definitions that link human phenotype ontology and gene ontology. Our next steps will explore ontology matching techniques to enrich links between ontologies, try additional ontologies and data, and include ablation studies to study the impact of extra knowledge. All data and code are available. Thank you for your attention.